In this video, we'll be discussing about the peptide bond formation and its properties. We see the peptide bond is an amide type of covalent chemical bond. It links two adjacent amino acids. We know during the process of translation, we get the proteins synthesized. And these proteins are made up of polypeptides. And these polypeptides are made up of peptides, which are linear chains of amino acids, as shown in the diagram. These amino acids are linked together by a bond called the peptide bond. Here in this diagram, we have the amino acid showing amino terminus and carboxy terminus. On the other hand, we have the same amino acid with same structure having amino and carboxy terminus. To get the linkage between two amino acids, the carboxyl group from one amino acid reacts with amino group of other amino acid. Here we see one loses OH from its carboxyl group and other loses hydrogen atom from its amino group. This reaction produces a molecule of water and two amino acids are joined by peptide bond. The two joined amino acids are called dipeptide. In this diagram, this is the peptide group and the bond between carbon and nitrogen is the amide bond termed as peptide bond. So what we saw in this reaction is the loss of water. Hence this process is dehydration synthesis reaction also. Now let's see some characteristics of peptide bond. The peptide CN bond is somewhat shorter than CN bond in the simple amine. So here this bond, which is denoted by the arrow, is a shorter bond. It is this peptide bond. Moreover, the peptide bond has a partial double bond character due to the resonance. And due to this resonance or partial double bond character, it adds up another property, that is the lack of rotation, as in double bonds. So the peptide group is fixed in either a cis or trans configuration. Now getting to the translation. We know it is the Peptidyl Transferase reaction which aids the peptide bond formation. Here in this diagram we have the two amino acids in the ribosome carried by transfer RNAs. One at P site and other at A site. It is by the enzyme Peptidyl Transferase which mediates the reaction. Here the nucleophilic attack occurs from NH2 towards the carbon atom of carboxy end. And by that reaction we get the two amino acids attached by peptide bond, which we have discussed earlier. And also remember, this peptidyl transferase reaction is mediated by peptidyl transferase enzyme of the ribosome. We see in prokaryotes it is the 23S component of 50S subunit, which has peptidyl transferase activity. Whereas in eukaryotes, it is the 28S component of 60S subunit, which has the peptidyl transferase activity. So this is the basic overview of peptide bond formation, its properties and peptidyl transferase reaction. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon and also make sure to subscribe this channel. Thanks.